The 2021 International Myeloma Workshop in hybrid format, both on-site in Vienna and online, was a great success, and great credit is due to the International Myeloma Society and the organizers for putting together the program and organizing the meeting under the overall challenging circumstances of the ongoing pandemic. The clinical, translational, and preclinical science presented at the conference consolidated several important concepts that have been emerging in the field in the last few years. And I would like to highlight some of these concepts because, in my view, they will play an important role in the near future. An area of extensive discussion at the conference was minimal residual disease assessment, MRD. As we know, there has been an ongoing improvement in complete remission rates in multiple myeloma treatments as new pharmacological and immune-based therapies have been developed, and especially since many of these therapies have been used in combination. Because of this progress, the assessment of minimal residual disease has now become a necessity for myeloma management, in the sense that it's broadly accepted now that targeting an MRD negative state should be the goal. So a key take-home message from this conference is that MRD assessment, which in the past could have been viewed as a luxury, now is a necessity. An important question that will be probably one of the most exciting translational and clinical research topics in the near future is if and how to apply MRD assessment in an adaptive manner to alter the therapeutic management of a given patient on an individualized basis. And there are several ongoing and upcoming studies that will provide formal answers to this question. Immunotherapies, of course, were a major topic in this conference. There were many updates for several important clinical trials for chimeric antigen receptor T cells and bispecific antibodies, including presentations on Teclistamab and Talquetamab that target BCMA and GPRC5D, respectively. These presentations consolidated the notion that bispecific antibodies are highly active, can achieve high rates of complete responses, even in patients that are heavily pretreated. The CRS rates with bispecific antibodies are lower compared to CAR-Ts and manageable. And so an interesting question from this conference is to what extent and how soon can we see some of these bispecific antibodies being tested in clinical trials in earlier stages of the disease, and especially in high-risk patients. Beyond the clinical trials on immunotherapies, there were several new immunotherapeutic concepts that were presented at the preclinical level. For example, one of them involved a BCL-XL armored BCMA targeting CAR-T cells. The group of Nizar Baylis presented this concept as a strategy to overcome exhaustion of CAR-Ts and enhance their persistence for the targeting of myeloma. From our own group, from my own lab at the Dana-Farber Cancer Institute, and in a collaboration of ours with the University of Helsinki, Sarah Gandolfi presented our collaboration on using CRISPR screens with single-cell transcriptome readout to understand potential mechanisms through which myeloma cells can be either resistant to or hypersensitive to natural killer cells. And we think from our own research that this is an important area of investigation, because it is poised to provide us more information about how we can individualize in the hopefully near future the uses of natural killer cell-based therapies for multiple myeloma. And this is an exciting area for us because our recent work in myeloma and other cancers has documented that natural killer cells can, in many cases, be active against tumor cells that have developed resistance to T-cell-based therapies. In terms of pharmacological agents, intriguing clinical data were presented in regards to the new category of cell modes. For instance, clinical trial data were presented for the combination of iberdomide with daratumumab, bortezomib, or carfilzomib in relapsed and refractory myeloma. And this is clearly an area of very active and very exciting investigation, given the clinical activity of the cell modes. From the basic science standpoint, many important areas of research were addressed in the conference. Single-cell molecular analyses of the tumor cells and their microenvironment, genomics of multiple myeloma, including the basic biology and translational implications of chromosome 1q gain or amplification. For instance, there were results presented from the FORTE and ENDEAVOR trials indicating that triplets involving a proteasome inhibitor and an IMiD appeared to be adversely affected to a different extent by chromosome 1q gain or amplification. There were also intriguing preclinical data on the biology of gain of chromosome 1q. For example, the group of Roger Tiedemann presented data indicating that chromosome 1q promotes mitochondrial oxidative phosphorylation, suppresses interferon response and tumor immunity in myeloma. And that was a very comprehensive analysis that enabled Roger and his team to look into the biological implications of chromosome 1q gain in a manner that corrects for potential confounding effects of other genomic lesions that may be co-occurring with a gain of chromosome 1q. Also, there was a lot of discussion about immunogenic cell death. For instance, Ana Maria Gula presented results about how the loss of function of a gene called GABA-RAP drives tumor cell resistance to proteasome-induced immunogenic cell death. Finally, from the perspective of a theme that is very important both for basic biology as well as translational research, there was extensive discussion about the use of bone marrow versus peripheral blood samples for molecular assessments of either genomic lesions or the molecular features of immune cells and tumor cells. And although it appears that at this particular point, the myeloma field considers that bone marrow aspirates still remain a gold standard, there is an increasing understanding that assessment of the circulating compartment, either cell-free DNA or circulating tumor cells or maybe even immune cells, could be considered a suitable or less invasive marker. But it is also understood and acknowledged by the field that more comparative studies, more formal comparative studies will be needed in order to understand in which settings and for which types of markers the assessment at the bone marrow or the assessment at the peripheral blood may be more meaningful. Overall, this was a very exciting conference for the myeloma field. A lot of excitement because of all the great new therapeutic options that are available for the patients and the continuous improvements in outcomes. Also very exciting conference in terms of the basic and translational science and how to make that science count for patients. Now, with all these options, there's more of a need for the basic and translational science to help us understand how to individualize the treatment for myeloma patients in order to be able to, in the near future, achieve ideally curative outcomes for as large a proportion of myeloma patients as possible. So a lot of excitement in this conference. Congratulations again to International Myeloma Society and the organizers for the excellent organization. But a lot of work remains to be done by all of us in the field. And I think our patients need us to make a lot of progress and reach curative outcomes for them as soon as possible.